This is a production of Cornell University.
So thank you so much for inviting me here to talk to you guys today. And it's been a great visit so far. It's really nice to be able to go places in person again. So I'm going to talk to you today about some of the work that Mike mentioned. So this work looking at sugar accumulation in sorghum and what structural variation has been doing in that process. So also, like Mike mentioned, I am broadly trained as an evolutionary biologist, population geneticist. And what I'm really interested in, sort of at the heart of everything, is just how do populations diversify? And particularly what goes on at the genome level that lets these things adapt to different places or take on a lot of phenotypic differences. And if these things are complex, if these kinds of changes are complex, then what does that look like at the genetic level and how can we find it? And so I haven't always worked in sorghum. I worked in all kinds of weird stuff, and even adding to the weird list of things I've worked on in the past, I've now got mosquito things going on and hummingbird things. So my Steve Kresovich used to call me species agnostic, which is a pretty good description. But I've been doing a lot of work in sorghum, and it's actually a really great model for sort of understanding this process of diversification. So sorghum is really phenotypically diverse, and we have all these different types, and I'll tell you a bit about that in a second. So I'm going to sort of give you sort of a case study here of a trait that we were interested in and how we started looking at a particular gene family because of what we found going on with these traits. So broadly, what I'll talk about today, I'll give you guys a bit of an overview of the history of sorghum and its diversification and what the different types are. And some of our past work where we looked at sort of global patterns of diversity and started finding things going on in metal transport. And then I want to spend more of the talk talking a bit about an actual functional study, looking at one gene in particular that was of interest, and it's probably the one molecular biology thing I have to talk about. And then looking a little bit, we looked a little bit at some expression stuff as well. So to give you guys an overview of sorghum. So sorghum was originally domesticated in Africa, kind of around where present day Sudan is. And there's some debate about whether or not there's a second origin. I feel like I'm gonna hit the wrong button for laser pointer. And then I'll give up. There we go. A second origin over here, but we know for sure there was one in this area. And then after that, it starts spreading east and west. And during this time, we started to get what are the different races of sorghum. So they take on sort of phenotypically distinct traits. You can see here where there's more humidity, you have these open panicles. Here, where it's drier, you get very sort of tight panicles with lots of seeds. There's also a lot of things going on with how they actually adapt to the climate and soils in these regions as well. So even more of this goes on as these spread actually away from Africa and into the Middle East to sort of China. So then actually they start coming back to these same dura types. They spread east, they then move back into Africa after they've already diverged a bit while they've been off the continent. And then finally you see things start to move south and you get this Kaffir race. And this is a big change because you're going from tropical to actually temperate latitudes with different day lengths. So during all of this time, you've got a lot of interesting adaptations going on. So sorghum is of interest to people in agriculture because it's very photosynthetically efficient, it's drought tolerant. You have it during its spread, it becomes adapted to all these different types of regions. What you can't see here is you're also adapting to different elevations, you're adapting to different moisture levels, you're adapting to different soil types. So we know there's got to be a lot of physiological and morphological changes going on. And so it does lead to the question, well, what's the genetic basis of that? And then on top of that historical sort of diversification, we've got things that are going on in the present day. So sorghum has been now cultivated for these diverse purposes where you can have types that you grow just for the grain production. They mature more quickly, they're shorter, they obviously put on a lot of grain. The types I'll talk about today are to these sweet varieties. They get a lot taller. They do put on grain, but they put on less grain. And the main thing is they accumulate actually a lot of sugar in their stalks. So you can press them like sugarcane. And that's where we get things like sorghum syrup and sorghum molasses. And then you've got these giant biomass types that never flower in temperate zones. They just grow indefinitely. Here's the top of a person. This is one of Steve's old pictures. I think this might actually be someone from Cornell who's in this picture. But just to give you some perspective of how big these things get. Yes, this is a. So I want to tell you a little bit more about sweet sorghum as well. So its sort of specific history, since that's what we're going to be talking about today. So sweet sorghum has actually also started to be cultivated pretty early. So even in the early domestication of sorghum, people are interested in actually getting sort of that chewing cane sweet varieties out of it. And so there's evidence for this going on, as early as 200 AD in that same region of original domestication. And then just like grain sorghum, the sweet sorghum varieties sort of spread along the trade routes. They go east and west and south. And there seems to be, there's now it's kind of more evidence that actually you have multiple origins of sweet sorghum. So each of those places that it ends up, some people might have started cultivating sweet sorghum from grain in those places. So that's kind of a parallel evolution of that type. And so, as I mentioned, what's phenotypically distinct about them is their height, the smaller grain heads, and the fact that they accumulate all these sugars in the stalk. And then, not necessarily key to the stalk, but sort of anecdotally interesting, sweet sorghum is actually the first type of sorghum brought commercially to the US. And it's because it's much less labor intensive than sugarcane. And so it was brought over by abolitionists who thought it would be an alternative to sugarcane, which required slaves. And then now we're interested in it as also a possible biofuel source, because you can actually use the sugars for biofuel. So if we look at just sort of the genome-wide genetic differences of sorghum, what we see is that their genetic differentiation reflects their history, not necessarily these present-day types. So these are several different studies. So this is using I want to say about 200,000 SNP markers. We've got about 1,000 genotypes. And what you can see with the clusters is that they very much genetically cluster with where they are from. This is a set of only bioenergy types. And they very much cluster by location. Here's a bunch of Ethiopian ones, and by that historic race. So this is the Kafir varieties, the southern varieties. Here's the original bicolors, and the Guineas, which are the western sorghum. And then more recently, we had a resequencing panel of about 320 accessions. We actually had enough data to do the structural variation analysis. So we looked specifically at deletions. And we can see that even in that different type of mutations, so these bigger, higher impact deletions, you still see the same sort of clustering. The font here is way too small for you, but it's again going by location and race is how things are dividing. So you can't just sort of look at the broad patterns of genetic diversity and compare, the sweets to all the grains and see what the genetic difference is. What you actually see is there's sort of historical difference. But there has been a lot of prior work done looking for what the genetic control is of sweetness. And so one of the key traits that people were able to isolate is actually stem juiciness. So this water content of the stalk. So here on the end, these are non-sweet varieties. They have what's called a pithy stem. And then here in the middle, we've got juicy varieties. And you can see when you dry them out, they look even more distinct. And this is actually a single genetic locus that controls this. It's called the dry locus. It was originally sort of characterized in 1916, but then no one had really looked at the molecular gene underlying it until 2018. So more than 100 years later. But they did find that this gene actually controls this transcription factor that regulates cell wall biosynthesis. And what goes on is the sweet sorghums have a loss of function deletion. And so what they get is much less stable cell walls, as well as some other changes. So it changes the thickness, and it allows you actually to accumulate all this water. And also, so you don't have these sort of strong structures even when you dry out. But even though that locus explains sort of your stem juiciness, it's not explaining all of the sugar accumulation. And in fact, this right here is BTX623, which is classified as a grain sorghum, and it's actually the current reference genome for sorghum. So it can accumulate some sugars, but it does not accumulate nearly the amount of the types that we see sort of pressed for the actual syrups or sugars. So this has a juicy stalk, but it's not actually all that sweet. Some other studies have tried looking more at just sugar accumulation in particular, with QTL studies, with GWAS, with sort of targeted candidate gene sequencing of things that we know transport sugar or things that we know synthesize sugar. And what's always been the case is you can find expression differences in the candidate gene in one line. The same thing is not going on in another. Same with the QTL. One cross has this big QTL in a particular region. Next cross you look at, it's not the same thing. And one of the sort of tricky bits about this, again, is we probably have this parallel origin of sweetness where there's maybe not one thing because it might have happened sort of multiple times in those different locations. And the other really tricky thing is that there's actually a lot of confounding from the maturity patterns. So the sweet sorghums take longer to develop in general than the grain type sorghums. And so they're also sort of accumulating sugar as they develop. And so a lot of times when you sort of look for differences, what you get are QTL that overlap the maturity genes. So with sort of all of the interest, sort of from an agronomic perspective in the sugar trait, and then kind of all these still unknowns about what was the mechanism of it, we decided to actually generate a reference genome specifically for sweet sorghum. Because one thing that we thought was, well, there might actually be sort of structural variation going on that would control this trait, and that's not something you would see by just taking short reads and aligning them to the reference genome. So while I was in Steve's lab at Clemson as a postdoc, we generated a de novo assembly for the Rio genome. So Rio is this sort of historically important sweet line that's been around for a while. And once we assembled it, what we found was actually Rio and BTX623 are quite similar in terms of their overall size, structure, and gene content. And in fact, this circle is, believe it or not, a Venn diagram sort of showing. So when you look at this in maize, right, if you're trying to show the amount of sort of gene content overlap, you get, like a circle here is maybe like 25% or 50% shared. Here, there's 98% of the genes are shared between the two lines. You maybe have a few genes sort of unique to each. 1% or less than 1% of the genes actually seem unique to the particular lines. Now, there's been a more recent study in the last year from Emma Mace's group in Australia where they actually did look at more of a sorghum pangenome. So they included more diverse lines and about 13 different sorghum lines and looked at this. And it puts it a bit into perspective for us. So what they find is there only about 36% of the genes are core, and then they have about 63% are what they call shell. So it's not in all the lines, but it's in more than one. And then a very small percent is actually unique to just a particular single line. And we have Rio here and BTX623. So what we know once we see this and we see this is that what BTX623 and Rio are sharing is a lot of the shell genes, as well as a lot of the core genes. So they have genes that they share with each other that other sorghum lines might not have, but that they do also share. And so we did go in and look at, well, they might not have that many differences, but they still got a few hundred. And so what were those? What genes does Rio have that BTX623 doesn't, and vice versa? And has that got something to do with the sugar phenotype? And we actually initially thought we had something really interesting. So we saw that a bunch of genes in this sweet transporter family were actually missing in Rio. So missing in the sweet sorghum, or not expressed at all. So they actually had enough of a mutation to be sort of completely, possibly non-functional anymore. And so we knew that these transporters moved sugar sort of out of stalks, out of leaves. And so we thought, well, maybe if you don't have these extra transporters, you just keep the sugars into your stalk, and that's one of the ways you can accumulate it. But this hypothesis sort of blew up. Once we got a lot more sorghum lines, we actually sort of did that structural variation analysis, and we tried to see, okay, are all these same things deleted if we look across a lot of different lines? And what we found is that no, it looks really random. So here I just sort of got broad categories. These are non-sugar accumulating tall lines. There's sort of a narrow thing of grain lines. Here's the sweet. Your purple and not purple are showing you where there's genes or not in certain families. And all I really want you to take away from this figure is that it's random. There's obviously not any sort of pattern here associated with how things are grouped on the bottom. So in other words, even though Rio had those sugar transporters deleted, if you look in other lines, some sweet lines have it deleted, some sweet lines don't. Some of them have other things deleted that Rio has. So it really just looks pretty polymorphic. So then we stepped back and wanted to think, well, okay, if it's not those things we already knew had to do with sugar, let's look at what's sort of broadly going on in terms of variation in gene content in the two genomes. And maybe that's actually where we should be more interested. So we looked at what types of genes tended to have presence, absence, variation in the two lines. And then also which types of genes tended to have really, really high impact non-synonymous mutations. So things that really look like they changed function. And what we see is that in both cases, your most significant categories are actually always related to disease resistance. And that's not that surprising. That's been seen in a lot of other plants. And it makes sense why that's something that you would have a lot of variation in gene content in. But one of these things that's always significant, although never the most significant, was metal ion binding or metal ion transport. And so we kept seeing it, and actually, particularly in your highly divergent mutation category, you see a lot of the metal transporters coming up here. And so it started to make us think, well, maybe that's actually something, because that was a little less expected than the disease resistance genes. And so we also did a differential expression study in the two lines to see if we could see which, in that sense, what things were going on. And I'm going to go through this a little bit quickly because I want to get to other stuff. But the general idea is that we have PR22, which was actually a recombinant inbred line from a Rio by BTX cross. And so what we did was we picked the one offspring from that cross that looked the most like Rio in terms of its maturity and it's all of its growth patterns, but just did not accumulate sugar. So we're trying to just isolate that one difference. And then we sort of looked at what the expression differences were between them. And then we also took the genes that were expressed differently and thought about, well, which ones actually are coming from a place in that RIL that had the Rio parent allele versus the BTX allele. And so we're most interested in genes that are very differentially expressed between the two and come from the non-sweet allele. So that would actually be the mutation within that gene that could be affecting the expression. And so what we saw was, in general, when we compare the patterns of expression, one of our most significant groups here is, again, sort of metal membrane transport. And what's notable about this group, and probably pretty hard to see from far away, is actually one of our only categories where it's more enriched for genes that have the BTX background than it is for genes that had the sweet background. And just to sort of reemphasize why that mattered to us is if you're very differentially expressed between the two, but you have the sweet allele, then it seems like you're just something that's responding to sugar, not necessarily something that's causing the sugar to accumulate differently. Because one of the things we know about sugar is that's once you're getting sweeter, you're actually signaling a lot of other pathways to change. So it's hard to differentiate. So we're not doing a real eQTL study here because we've got one thing. We're just sort of doing a very poor man's QTL where we've got one line and we're just thinking about the genetic background in it. And then we went back to our genes that we knew had really high impact mutations, and we tried to overlay that data with what was actually very differentially expressed between the two. And so we're looking for things that have the predicted functional effect mutations and look like they're expressed differently when you have the genes on the two different genetic backgrounds. And we get a handful of things that are actually known, not just metal transporters, but actually really known to be in the iron transport pathway. So we have some ZIP genes, we've got a YSL gene, and then we also got these BH-LH transcription factors, which are known. Now there's a big family, but one of the main regulators of iron transport, one of these transcription factors, is actually a BH-LH type transcription factor. And then kind of the final past study I want to quickly tell you about. So I had mentioned that we started looking at a broader panel of about 300 or 400 individuals, and we had done resequencing at about a 50x step so that we could look at structural variation particularly deletions. It's pretty hard to look at structural variation with short read data, even with a lot of it. But what we saw when we looked at genes that had a lot of actually large deletions, so 50 base pairs or bigger, we saw that if you isolate them down to the deletions that seem like they're happening in a specific population or a specific group, then also you see metal transporters very much enriched in that group. So there's no way you guys can read this, but here, the colors here are sort of showing you for the deletions, which ones are happening in sort of the different groups. So the colors are your different populations. Here we've actually got some gene IDs. The only thing you can really take from this is that the place where we actually see the metal transporters seem like they have the most deletions is in the red group. And the red group is specifically over here in Ethiopia, and it seems to be the Ethiopian highland sorghum. So it's a particular group of them. So not necessarily related to sweetness in that case, but still showing something about sort of population adaptation and diversification in sorghum. So I do want to take a second here. So I showed you these sort of very short, quick snippets of a bunch of different studies we've done over the years. And one thing you might have noticed from them is that we see metal transporters as significant, but it's never the most significant thing. And there's a lot of other stuff there. And so you might ask the question, well, why did you guys zero in on this metal transport thing? Because it's certainly not the only thing. There's all kinds of disease resistance and other types of genes. And one reason was just because it was one of those categories that it just kept recurring, kind of no matter what study we were doing, and we were never really looking for them. We kept seeing something related to them. But then the other reason comes from this study that I'm about to show you next. So while I was doing the work on the Rio genome, there was a graduate student in Steve Kresovich's lab who is looking at genome-wide association for sort of bioenergy related traits. And one of the traits he was looking at was stem sugar content. And he found a very significant GWAS hit sort of here on chromosome 4. And then he redid the study and he found it again. And what was under that GWAS was actually this particular vacuolar iron transporter, or something that was annotated as that. And when we first saw that, we just didn't really know why that would be coming up for this trait, but it kept being the most significant thing that we were seeing. And so if we look a little more closely at the haplotype, what we see is there's a really strong haplotype structure where if you're sweet, you have this, you almost always have all the blue alleles. And if you're yellow, you've got this. And there's a very significant sort of grouping in like how much carbohydrate content you have, depending on what haplotype you have. So the region itself, it's GWAS, it had 4 genes. And only one had the non-synonymous mutation, and it was this VIT, or this vacuolar iron transporter. So it was kind of a good candidate, but it wasn't a great one because the non-synonymous mutation was actually not in the functional domain. It was in some domain of the protein. We didn't really know what it was for. It was originally annotated as nodulin-like, and sorghum doesn't have nodules, so we really didn't know what to make about that. The public expression data, it looked really low, so a lot of things made it look like maybe this wasn't actually a real gene. It might be a pseudo gene. And then it didn't overlap with any of the QTLs sort of previously identified for this trait. But then, so a new version of the reference genome came out, and I created the sort of Rio reference genome. And when we looked at this region again, it actually started to look more interesting, because what we could see now was that what you had was a tandem duplication in the vacuolar iron transporters that only happens in sorghum. So everybody else has this one copy. Sorghum has a second copy. Here's where our GWAS candidate gene was. It's in that sorghum-specific copy. And then what was additionally really interesting is when we looked at the Rio assembly, we actually saw that there was a lot of difference. So you had a lot of non-synonymous high-impact mutations in this gene, and actually the Rio gene was much bigger. So the original reference genome, the BTX gene, looked like it was truncated compared to Rio. So actually it looked more like a real gene when we looked in the Rio genome. And so we had a collaborator at Penn State who actually helped us with the molecular characterization so that we could actually see whether or not this really transports iron and whether or not there's a difference in what the sweet and non-sweet alleles were doing. And you guys probably understand this better than I do. But so what they did was they had a yeast strain that was iron sensitive. So the transporter had been knocked out in it. And they transformed it with an empty vector, the Arabidopsis bit that we know exactly what it does. And then the two alleles, so the sweet sorghum one and the non-sweet sorghum bit. And then you grow it under different iron conditions. And the yeast can only survive if it's able to transport iron, because it's got to be able to pump it out. And what we found was that only the sweet allele for sorghum shows that it can transport iron. So only the yeast with those alleles are able to survive. So in your empty vector, under control conditions, they grow, but you add iron and they don't grow at all. Here's Arabidopsis, where it can grow in both. The Rio is the sweet allele. It can also grow in both, but the BTX, you only see growth in the control, sort of no iron conditions. And that's what I was saying. So that was actually pretty interesting to us, because it suggested, one, this thing functions as how it was annotated, and there really is a functional difference in the two types. And so there seems like it really could be doing something important for these lines. But it's still not entirely clear what it has to do with sugar accumulation per se. So we also wanted to sort of say, well, are sweet sorghums better at dealing with iron deficiency or iron stress than non-sweet sorghum? So maybe they can grow in lower iron conditions, but still make enough sort of sugar or photosynthesize enough. And maybe that's one of the ways they're getting sweeter. So what we did was a pretty straightforward sort of pilot expression study. So we took about 3 genotypes of each type. We grew them in a hydroponic environment in different iron conditions. So we had control and then iron deficient. And then we just wanted to look at expression differences. And we were looking in pretty early stage in the roots. And one of the reasons is because the evidence we did have for expression of that VIT1 was potentially that it was in the roots of seedlings. The other reason is because these sorghums get really big, and we didn't want to do that in a growth chamber. And so what we saw is that you get a lot more differentially expressed genes in your control versus low iron conditions, sort of for everybody. So that these conditions of just your being an iron deficient has a big effect on all of the genotypes. You still have a fair number of things just differentially expressed between the two types of things, even if they're in the control conditions. But then you do get a handful of really interesting things where there's a significant interaction. In other words, the way the different types respond to the iron deficiency is different. And just to give you guys a little bit of background on how we actually did the analysis, we did a couple of things that weren't entirely standard. So one thing is, we had these multiple genomes now. So we actually tried mapping our data back to both. And what we found was that in general, because everything we're growing here is either biomass or sweet type, they map slightly better to the Rio genome than to BTX. We get a higher percentage. So we went forward with those results. And we were interested in these things. We knew that one gene was a tandem duplication. So we knew we were interested in things where you actually might get multiple mapping hits. So we used a program and a method where you would retain those instead of discarding or keeping one or the other, which is sort of your default method. And then we also, we're trying to look at type here, but type is just sort of genotype is a subset of type. So we have to look at difference between types and control for differences among the genotypes. So rather than your standard expression analysis, you treat each thing as a factor. What we actually did was treat your genotypes as a random effect, kind of like how you would do a GWAS. And so what we found, and I'll start with the middle bit. So if you look for your enriched kind of Go terms in your low and normal iron conditions, you actually get a lot of the things that you expect. So there's things related to iron transport and metal metabolism and iron stress. And there's a lot of known orthologs of like NRAMPs, ZIPs, COPTs. So those show up as significant in this comparison, but they are significant regardless of what type you are. So that's just kind of your conserved response to iron. The things that show up kind of in your differences between types are maybe less obviously sort of related to the different, to whether or not it's iron conditions. So it seemed like different types of things. You actually see sort of a lot of ADP binding, some carbonate, dehydrolase activity, things that look a lot of phosphatase activity. That's what's really going on in here. And similarly, over here, when you look at the things with significant interactions, it's more likely to overlap with the categories for your differences between types than necessarily for the category here. Although there are a few things where you've got known sort of iron transport genes and they actually respond differently in the types. So just to give you guys a couple of examples of some of the more significant things we found. So here is one gene that's a transmembrane transporter. It's not actually a known metal transporter, but it is in this nodulin-like transporter family. And what we actually see is that in your non-sweet varieties, when you expose them to the low iron conditions, it actually gets slightly downregulated, whereas in your sweet varieties, it becomes upregulated. So the level of expression actually shifts. Whereas this, so here's this BHLH transcription factor that looks like a homolog of these IRO2 or IRO3 sort of known regulators of iron response. And you can see it's very much upregulated in both the groups. It looks a little bit steeper in one, but I think that's actually sort of an artifact of how much variation you have, not necessarily that it's really. So this is really something that shows up in both groups. It's not actually different between the types. And so this last analysis I want to talk about, one of the things we noticed, so the Go enrichment didn't really tell us that much, but one of the things that we noticed was that when we were looking at these differences between types, it seemed like a lot of the time it was sort of coming up in genes without a lot of known homology or known function compared with other species. So we actually wanted to explicitly look at that and say, are differences between types more likely to be happening in genes that have a more recent evolutionary origin in terms of their copy number? So we did an orthology analysis across a bunch of representative species. And what we actually did was try to determine for every gene and all the genomes, what was the origin of that particular gene copy and put it to a branch on the sort of known tree among all these organisms. And then once we categorized all the genes, we saw, well, which ones of those, of the differentially expressed, where do they tend to be on this tree? And what we find, if you look at your differentially expressed genes, again, for all individuals between your control and low iron conditions, you don't actually see particular enrichment at any one node. You kind of see it's more or less distributed how you would expect just based on how the genes are distributed across that tree, that history of the tree. You have a little bit of enrichment maybe in grass-specific genes, so genes at the origin of duplication of all the grasses, but that are shared among them. But if you look at your differentially expressed genes between just your genotypes, it is very much enriched in the sorghum specific or sorghum polymorphic category. So it's really going on. It's very enriched in this sort of new genes, and it's significantly depleted in things that are shared among all angiosperms. So there's really conserved genes. You just don't see a lot of difference among types, or you see less than you would expect by chance. It's not as obvious when you look at the interaction genes, but you do have some enrichment here in kind of your unknown ortholog genes. But again, what you really see is it's never going on in those really conserved genes. So it seems like there is something to do with sort of having these new copies that lets you get these differences between the types. So I feel like I went through really fast. Okay, so to start sort of summarizing this, so we know that this sort of metal binding and transport show a lot of variation, both in terms of their presence and absence and their sequence divergence. When you look between just different lines of sorghum, and we know that some of this seems like it's connected between the difference between the sweet and the non-sweet types. Some of it also looks like it might just be connected to just kind of global sort of adaptive differentiation. We don't really know what the direct link is between the metal metabolism and the sugar accumulation. We know that iron uptake is pretty crucial for a lot of functions in plants, including photosynthesis. So it could just be that if you're better at taking up iron out of the soil, you're just better at producing sugar or whatever product you're trying to make. But one alternative, and what I tend to think is more going on, is that your different copies of the different genes might be getting specialized functions. And so it could either be transporting things at different times, or possibly transporting molecules other than iron. And we also saw that when you compare the types in terms of expression, it looks like your newer gene copies are more likely to be linked to the sort of type-specific differences. And the older copies have maybe maintained their original roles in the sort of stress response that everyone has. And then we know, at least in one case, we know that we have this sorghum-specific vacuolar iron transporter where it has a loss of function in your grain type. It still has the function in the sweet type. What I didn't really get into is that loss of function. It doesn't necessarily have to be loss of ability to bind or transport iron. It could be loss of ability to get localized to the vacuole, because that would also mean you can't pump things out from where you're supposed to, and that would also let your yeast sort of die from that. So we still have some questions about what function is it actually losing between those two types of things. So I flew through that, and I'm sorry. But I do want to stop and thank some people. So my former postdoc, Kitty Kuhn, he's actually a graduate student from Cornell. He did all of that sort of global structural variation, deletion analysis. And he also set up the RNA-seq hydroponic study and did a lot of that work as well. My student, Azita, started in on the RNA-seq analysis and did a lot of that differential expression. And then as well as my former undergraduate student who had the pleasure of doing a bunch of RNA extractions and taking care of plants in the growth chamber. And then my current students, Will and Bailey, have sort of been the ones who've taken over some of this work. And then I also want to specifically thank some of my external collaborators. So Erin Connolly at Penn State is the person who actually did those yeast complementation studies. Actually, probably her student Brandon is the one who really did them, but they happened in her lab. And then Jeremy Schmutz from Hudson Alpha. So their group did a lot of the Rio sequencing. So we've worked a lot with them on that. And then Doreen Ware at Cold Spring Harbor, we are currently working with to do some new genome assemblies specifically related to the lines that we tested iron in, and also do the annotations for those and get them into a database. Then of course, I want to thank people from my former lab at Clemson, where I did a lot of that earlier work related to the GWAS and first assembling the Rio genome. And with that, I am happy to take any questions.
So the VIT1 and VIT2 are both non-functional in the grain type sorghum?
So the original VIT copy is functional, but that second sorghum specific copy in the grain sorghum, it looks like it doesn't move iron out of the vacuole. And like I say, it's not clear if it's doing that because it can't move iron or because it's not getting to the vacuole to move it. But it is definitely truncated compared to the other allele at that same gene copy in the sweet sorghums.
Okay, and you don't have a mutant of the other bit then?
Of the
in the grain sorghum.
Oh, you mean we haven't put in, we haven't tried to transform with the?
Yeah, you do have a knockout of those. You know, the one's not functional, but the other one is, and I'm wondering if you thought about knocking the other one out in grain sorghum.
So we have thought about doing, well, we thought about, we actually put in a proposal where one thing we want to do is actually at least try it in Arabidopsis, so try it in plant rather than in yeast. But no, we don't currently have a knocked out copy. And it's, you know, just like you say, it's a little tricky because we know everybody's got one functional copy. Right. And so I think the trick is trying to see what just this extra copy might be doing.
Thanks.
Do you see diversification of the expression in the functional paralog in the sweet sorghum?
So we've had a really hard time. You know, one of the disappointing things about the RNA-seq experiment we did is that we didn't actually see expression of that particular gene at all. And we've had a really hard time figuring out when and where it is actually expressed. And it doesn't seem like it's at the same time as that original sort of gene copy. So we don't quite know where to find it. We had sort of seen in public data, we saw this sort of like low bump of expression of it, you know, possibly in those early root seedlings, but now I think maybe that wasn't real, or maybe that was stuff that should have mapped to the other one. So we're still trying to figure out where it is.
I think maybe just new genome assemblies.
That's a good question. I think one thing that it might help with the new assemblies, particularly with the de novo annotations, is we might get a better sense of, is it super polymorphic in terms of who has what looks like a functional copy or not, same as some of those sweet transporters, and that might make us less interested in it if you don't sort of see this, all the others conserving what looks like a functional allele. In terms of sort of finding where it is, hopefully if people do a lot of iso-seq to do their annotations, the more data we get from more time and tissues, the more we might look up on it. I do kind of have a sense that maybe you might need it under stress to actually see it. Yeah, the right stress and then the right tissue, and then the right time. Yeah. So, yeah.
Different races of sorghum at the beginning. Is all your work that you were describing, is that in one particular race, or are you seeing this metal, this iron, and all?
So we see it, we actually see it in all the different ones, and we try to, in all of our studies, we try to include sweet and non-sweet types that represented multiple races, so that it wasn't just sort of all one versus the other. But I will say that what you can breed the types from really any different race, but if you look at all of them, there's a tendency for the sweet types to sort of come from the Eastern varieties or even that original bicolor race, whereas the bioenergy types might come more, you might see also sort of the Western sorghums. But you can see some of each. And so we picked our panels to try and catch that as best we could.
I was curious if you had any idea if like other high sugar accumulating crops like sugarcane itself, if they exhibited any sort of, weirdness and something to do with metal transport.
So I don't know if they exhibit that, but what I can tell you is that in sugarcane, they've looked more at the, these sort of targeted expression studies, and they've had a lot more luck where you can really see differences in known sucrose synthase that you can relate to sugar accumulation, and that has very much been selected for in the domestication process. And so I don't, I don't know, that's sort of hard genomes. I don't know what they've looked at in terms of the whole genome levels of variation or variation expression. It'd be a good question if they also show the metal variation, but they definitely show more obvious variation in some known pathway stuff. Yes.
You had a cool slide observation. Thanks for your talk, by the way. Of these genes that are new at different levels of the phylogeny, and then whether they're differentially expressed between iron or control, or between sweet and non-sweet. And I wondered if can you sort of extrapolate from that what the general the general hypothesis around that is, like if I were to go to some other traits, maybe tall and short, or maybe, I don't know what, just, different kinds of differential expression, or instead of iron deficiency, shade, or something like this, what would I find? Like what's the bigger framework that explains what you observed there?
Yeah, so that's a really good question. So I would say sort of the longstanding evolutionary theory would suggest that in stress or environmental change related things, you might be more likely to see diversification in newer gene copies. Because what happens is you get through some sort of genome duplication event or some other mutation, you can get extra copies of some of your genes. And then a lot of times, plants will go back to diploid, or what they'll do is they'll sort of lose extraneous copies. But while you have those extra copies, you've now got relaxed selection on them. So I've got the thing that works, and now I can do whatever I want to this stuff. And a lot of times it's going to be a pseudogene, but that it really sort of makes this substrate for this neo-functionalization of genes. And so one theory is that one thing that would drive sort of rapid neo-functionalization is disease pressure. But another one would be something like environmental stress. And I think I was telling Kelly earlier, they've looked at some kind of broad correlations between, these known angiosperm whole duplication events and retention of gene copies, and some triplications in some of these other kind of known orders of plants. And they seem to coincide with kind of moments of like global climate shift when you might need an environmental change. So to my mind, one thing is I think you could have natural variation sort of going on in newer sorghum copies of something like a metal gene or another stress-related gene, and they've been selected to, one, keep extra copies, so these families are bigger, and then selected to be able to sort of deal with slightly different situations. But I can imagine that in human, when we start selecting on things in artificial selection scenarios, that might be the same substrate we inadvertently go to because you know it's not going to kill the plant. If you're looking at these newer copies where you have a redundant copy that's still doing the old job, right? So you don't want to mess with like a developmental gene, but if you've got some of these extra copies and things that aren't constitutively expressed, but expressed under stress, that's a place you're more likely to see divergence. Does that make sense, or did I make it more confusing?
Well, I guess I'm sure that makes sense. So there's a question of how smart I am to be able to take your answer in, but you see one phenomenon for iron deficiency versus control, and a different phenomenon for sweet versus non-sweet. Your answer here had to do with stress, which made me think, okay, well, why did the iron versus deficiency versus control show this thing?
Oh, I see. Yeah.
But
yeah, I see what you're saying. So I think there's kind of a gradient of age. So I think one thing we did see, and it's actually not as significant as I thought, but it's slightly enriched in your GRAS-specific genes. You know, what's going on in your iron deficiency versus not. It's definitely not enriched in, angiosperm. Everybody has it. And so I think that it's slightly newer copies, but maybe lots of species have had to deal with some metal stress in general. But sort of the more recent sorghum diversification where we moved it around rapidly to lots of places, those are the even newer genes where you see changes. But I think what you bring up is a really good question. It certainly would be worth looking. It'd be nice to look at some expression data and just see, possibly if you even take public data, how many types of studies would you see enrichment in a particular sort of phylogenetic category to see whether or not, it does seem like things to do with human selection versus stress selection, that sort of thing.
One question on.
You want me to read it? I can.
I got it.
Okay.
Oh, it just says okay. See, do you have information about metal contents between the lines? Oh, no. That's a very good question, and that is something I just put in a proposal to try and look at, because that's something that we definitely want to know. If, rather than just looking at the activity of the transporters, is do you actually see a difference in, who actually has a bunch of iron in them or not? If hypothetically you think one is better at uptaking it, but it's not something that we have seen yet.
Yeah, I think early in your talk, you mentioned that a lot of the different differentially expressed genes were in response to pathogens, maybe. I'm wondering if the VIT1 could fit into this scenario, just because in rice, the bacteria that causes bacterial blight activates a sweet in order to fuel itself. But there's a paper a while back that showed that the COPT transporters for copper can actually bind to this sweet and reduce pathogen spread. So I'm just wondering if there might be some sort of shared molecular mechanism here of creating reactive oxygen species or something that might.
Yeah, I think it's a really good point, and I think it's definitely possible. And that what's actually going on. Even though it can move iron, that maybe that's not its primary function or not its primary role in terms of what would make the types different. And I completely agree that I think it could be something to do with communicating, either signaling or, like you say, sort of pathogen response. Because certainly metals play all kinds of roles in that, and then even as like Miguel and I talked about, things that are called metal transporters don't have to only transport metal. And so, but then if I start thinking about that, I might just kind of despair as to what we could ever figure out. So, yes.
Good question. So clearly there's huge differences in sorghum between in the amount of sugar that can stay in stems. Is this a general thing in grasses, like within a genus or within a species of variation in sugar or maybe starch content, or is this like sorghum or maybe, I don't know, sugarcane specific?
So I think, they have sort of maze lines that can also accumulate sugar. I suspect there's probably mechanisms in grasses where it's possible to at least evolve this, or there's some underlying machinery that can get selected on, and that's happened multiple times. And that's why you can see it in sugarcane, see it in sorghum, and see it in maize. But in terms of like what we've actually selected for to accumulate sugar, I don't know if those variants were kind of in like an older ancestor, or if it's just the machinery, and then you've got a highly duplicated region or a complex region where it's easier to get new structural variants and then do this again, and that's why you would see it. But one thing I will say, and I kind of mentioned, is that at least between sugarcane and sorghum, the actual mechanism looks different. The first thing people did was try and look at the sucrose synthase. It's just not, like I say, you can see difference expression, but if you try and look at different lines, the different types are just doing totally different. It's just all over the place. So it's not really fundamentally underlying it.
But so like rice, is there sweet stem rice and non or wheat, or I don't know anything?
I don't think, not that I know about, not in a way where it's ever been sort of commercially produced to do anything like a biofuel or a syrup. But there might be something, someone might have like a rice syrup somewhere that I don't know about, but I don't think so. And for those, when they do the alcohols, I know it comes from the grain, which the sorghum alcohol, sometimes they do it from the grain, but you can do it from the stem sugar as well.
Well, this has been a fun Q&A session. So thanks for doing a lot of time for that. For those of you who need to leave now, you can leave, or if you want to hang out for more Q&A,